Sounds good. All right, well, happy deep day, everyone. So excited to be here this morning or afternoon or night or insanely early in the morning, to be here as we celebrate the deep ocean and all the ecosystem services that it offers us. We have an amazing panel today, and our session is called Endless Discoveries, Unveiling the Marvels of the Deep. And let me just jump into some quick bios of our speakers so that it's less of me talking and it's more of them and their lovely presentations. So first we have Dr. Lissette Victorero, who is a deep sea ecologist at the Center for Coastal and Marine Research of the Norwegian Institute for Water Research. She also serves as science advisor for the DSCC Global Seamounts Campaign. We have Dr. Michelle Taylor, a deep sea researcher at the University of Essex with work centered on deep sea corals. We have Sadie Mills, and Sadie couldn't join us live today. She's based in New Zealand, and it's a little bit early in the morning for her, but she did send us a video presentation. And Sadie is the collection manager of the New Zealand National Institute of Water and Atmospheric Research invertebrate collection, and she was part of an expedition that recently discovered over 100 species thought to be new to science off the coast of New Zealand. And lastly, we have Salome Buglass. And Salome is a PhD candidate at the University of British Columbia, studying seamount habitats in the Galapagos. And that's it for our speakers. So I would just like to say thank you all, our amazing panelists, for joining. Everyone here excited and ready to learn, and we could dive into our first presentation, which is by Dr. Lissette. So the floor is yours.
Great, thank you, Khadija. You can all see my presentation, right? So, hi everyone.
Sorry, you need to share your screen again.
Let me see over there. Share. Okay, are you seeing my presentation now?
Yeah, but you need to put it on presentation mode, I think.
Oh, it's the other way around. Is that right now?
Yes.
Okay, great. Okay, so hi everyone and happy Deep Sea Day. Thank you for joining us to this event. So my name is Lissette Victorero. I'm a deep sea ecologist focusing on complex ecosystems such as seamounts and hydrothermal vents. And I'm really passionate about deep sea fisheries as well. So today I'll be talking to you about the deep ocean and the diversity within it and why the deep really matters to our well-being. My talk is titled A Deep Sea Planet. And the reason why I called it a deep sea planet is because when we contemplate the vastness of our planet, it's imperative to recognize the sheer magnitude of the deep ocean. It constitutes approximately 70% of Earth's habitable space. And so, as such, the deep ocean truly represents the last wilderness on Earth. It is a realm of mystery and intrigue that for us scientists, and I think in the future, it will be the same for the civil society as well. The deep ocean commences beyond 200 meters. You can see here in this figure on the right, much like the stratified layers of the atmosphere, the deep ocean delineates itself into distinct zones. Each of them has its unique characteristics. From the mesopelagic to the abyssopelagic, these zones extend to depths reaching a staggering 11 kilometers below the ocean's surface. To put this into perspective, this depth exceeds even the summit of the tallest terrestrial peak, the Mount Everest. The immense depth gradient, coupled with the expansive volume of water and the seabed area, creates an habitat of unparalleled diversity. Within these depths lie a myriad of ecosystems, each teeming with life forms that have adapted to the extreme pressure and conditions of the abyss. Within the deep ocean, there are many different habitats, each contributing to biodiversity and ocean health. Some of these habitats may seem familiar. They resemble those found in shallow waters, such as the vibrant coral reefs and the lush sponge gardens. Also seamounts, they rise as underwater mountains, similar to what we see on land. But in the sea, they act as biodiversity hotspots, teeming with thousands of species. Then we have the abyssal plains, vast expanses of sediment, where high biodiversity flourishes. But amidst the familiar lies the extraordinary. The deep ocean harbors environments of extreme conditions, such as the hydrothermal vents, the whale falls and cold seeps. Here, specialized fauna thrives using chemosynthesis as their energy source. These habitats are so unique that they push the boundaries of what we understand about life on Earth, but it also makes us think about what is life on other planets like. And last but not least, let's not forget the trenches. These are the deepest points on our planet that we understand basically very little about. They are the true final frontiers of exploration. Collectively, these diverse habitats, coupled with the vast depth gradient of the ocean, form the backdrop for an astounding array of biodiversity. Yet, physically accessing the deep ocean for us researchers is really challenging. The sheer size, the remoteness of the deep ocean, and coupled with the technological and financial hurdles of exploration, it makes the process of sampling and discovering species, in fact, quite challenging. Deep sea biodiversity, in particular, remains a realm of boundless exploration at the moment. The unknown far outweighs the familiar. Consider this, scientists estimate that over 90% of the species inhabiting our oceans are yet to be discovered. If we narrow our focus to the deep ocean, well, it's there that our knowledge becomes even more humbling. We've barely scratched the surface, having sampled less than 1% of the deep ecosystem. Yet, even within this minute fraction, we've made remarkable discoveries and discovered 400,000 species or more. But when we think about the implications of this, if we've uncovered such a wealth of life with just a cursory glance, how many species are out there in the deep ocean left to be discovered? Scientists speculate that the deep ocean may harbor anything between 5 to 10 million species, that they're just awaiting to be discovered when we have the resources to do so. And I think today, you'll see later how basically just the 20-day exploration has already uncovered hundreds of new species of New Zealand. So this brings us to what is the significance of the deep ocean for us, for the humanity, basically, and why is it crucial? Most people do not interact with the deep ocean directly. It is not very obvious how this realm provides benefits to humans. So here's a figure that we see there's an array of ecosystem services provided by the deep sea, including climate regulation, food sources, and biogeochemical cycling. All of these ecosystem services directly and indirectly benefit human well-being and the health of our planet. If we take, for instance, climate regulation, the role of the deep sea is really, really important. The deep ocean serves as a vast reservoir, absorbing and storing carbon dioxide from the atmosphere. It helps us mitigate the effects of climate change that we are currently experiencing, and the deep waters and sediments are responsible for storing billions of tons of carbon away. This crucial function stabilizes Earth's climate, but it also safeguards the future of generations to come. If we look at the deep sea biodiversity, why is it important? When we think about, let's say, sponges, they boast remarkable biodiversity in the oceans, and in the deep sea, they provide hotspots of biodiversity for other invertebrates. But for humanity, they also have significant benefits. They hold a lot of biotechnological promise. Their compounds are being explored currently for pharmaceutical applications, such as anti-cancer drug. And the silica-based skeletons of sponges, which you see in this figure, you're seeing a beautiful skeleton. They offer innovative avenues in tissue engineering and regenerative medicine. And this is very exciting, and there's so much to be found in the future as well. As I wrap up, I'd like to leave you with this powerful quote. The ocean's beauty is in its clear waters, but its strength lies in its dark depths. Thank you all for your attention and participation in this great event, and I can't wait to hear the rest of the presentations.
Thank you so much, Lissette. And what an amazing way to end your presentation. And I just love learning about the deep ocean. I just feel like every time I see a presentation, there's just so much new things to uncover, to just like learn, connect with. And I also love the fact that you mentioned the importance of the deep ocean in climate change. I know sometimes the deep ocean is something that is very much out of sight, out of mind. So appreciate you bringing that to life for our viewers here today. And just a quick reminder that this session is being recorded. And if you have questions, you could use the Q&A function and we would get to that at the end. And our next speaker is Dr. Michelle Taylor. So Michelle, floor is yours.
Thank you very much. Good morning, good evening, good night, everybody. Yeah, we've got a global audience here. All right, hopefully you can see the screen. I have a little thumbs up. Is that okay? Fabulous. Well, today I'm going to be speaking to you about deep sea species discovery. So my name is Michelle Taylor. I am a senior lecturer at the University of Essex, and I'm also president of the Deep Sea Biology Society. And so, yeah, the deep sea is my bag. Specifically within the deep sea, my favorite things are corals. Now, I'm going to talk to you about species discovery, but because they're my favorites, I'm really going to talk to you about corals. Scleractinia are the things that make the shallow water coral reefs that a lot of people are probably way more familiar with than the deep sea coral reefs, because there are deep sea coral reefs. Like in the shallow water, in the deep sea, it's these Scleractinia that make the reefs, but there's only about 16 to 20 species that make reefs in the deep sea. But there's, in total, including all depths, there's 1,680 species-ish, because new ones found all the time, and about 50% of them are in the deep sea. So in terms of diversity, there's a lot of diversity down there in the depths that Lissette has just beautifully described to us. Now, octocorals, Scleractinia, hard corals, they're okay. Octocorals, the soft corals, those are my favorite corals. And the deep sea holds so far a staggering 75% of the diversity that's found. Now, one of the reasons that I think corals are amazing is because if we just zoom in a little bit, you can see that they're a habitat. So they're a habitat for loads of different types of species and animals. There's anemones in the far left-hand corner of this screen. There are snake brittle stars dotted all over the place. There's probably God knows how many polychaetes. So crawling across this beautiful bubblegum coral. Now, the bubblegum corals can live for about 400, 500 years old, we think. This one was probably, this is a Southwest Indian Ocean one, and it's probably about 3 meters tall, quite tall. Some of them that we found in Antarctica, that are just about the size, less than a foot tall, they're over a few thousand years old. So corals can actually be some of the most aged things in the deep sea. There's been one that's been aged at 4,500 years. So they're actually some of the oldest things that even exist on our planet. So I find them extraordinary for many reasons. Now, they are also not very well, in terms of their distribution, we don't have a very good idea of the range. For example, if we were to think about one species, does it occur across the North Atlantic, or does it occur all the way down to the South Atlantic? We don't have a very good grasp on that yet. We do for some areas of the world. So I'll show you this map, because every dot on this map is a different type of coral. And these dots represent where we found them. Now, what becomes obvious if you look at the map for a short period of time is that we've done quite a lot of finding of coral in the North Atlantic and around American seaboard. Like there's the very well visited sections of Antarctica on the peninsula. That's not bad. There's some really heavy spots around New Zealand, because they've done an amazing job of exploring their economic exclusion zone. And the economic exclusion zone is, if you see the land masses, is that line around all the land masses. That's kind of what's within the jurisdictions of a country. Outside of that is the high seas, where there are less rules and regulations, and also it's less explored, but it's all deep sea. So you can see there are big gaps in our global map of where we know where corals are. So big gaps in the Pacific, big gaps in the South Atlantic, and really big gaps in the Indian Ocean, which doesn't have very well funded deep sea programs on its shores, like, for example, the North Atlantic does. So the Indian Ocean specifically is an area that I'm particularly enthusiastic and passionate about. And we were lucky enough to go on an expedition. So normally in the deep sea, this is kind of step one for a lot of species discovery, is actually getting out there and seeing what's there and collecting some samples. So not just taking images via a video, but actually getting samples in our hands. So in 2011, which is a very long time ago, we were lucky enough to go on the Royal Research Ship, the James Cook. It was 51 days at sea. There were 19 scientists on board, 12 technical crew who helped with all of the amazing technology that we used on the expedition, and 34 ship crew who essentially kept us all alive and well, and we couldn't do it without them. And where we went were the 5 seamounts that are black dots on the map that you can see. So we started in the North Atlantis, middle of what, Satma, Melville Bank, and Coral Seamount. Now, all of these are seamounts. So from the bottom to the top, they tend to be over 1,000 meters tall, and similar to seamounts on land, they're mostly hard, right? So they're hard substrate. And that's important for coral, because most coral, when they land, and they start growing tiny little polyps, they try to grow tall enough to get into the water current so that they can feed. So they filter feed. You know, when you see your beautiful David Attenborough programs and there's all of the marine snow falling down, that's what they're eating. So that's what they're taking up inside their bodies and making skeletons out of. So they're trying to get themselves tall enough to be able to filter feed that from the environments. And they need a hard substrate usually to do that off. So that's why they're important. So we were lucky, we're on this ship, we're, you know, almost 1,000 miles away from land, and we're putting into the water some remote operated vehicles, which are like robots, which have got brilliant cameras on the front and some manipulator arms to pick up items that we would like to see. Now, this is an image taken from that remote operated vehicle, and this is what I would call an octocoral garden, right? It's got such a diverse array of things in front of you. There are a lot of yellow fans here. Now, not all yellow fans are the same species. Just having a little look, I can see, I wonder if you can see my screen, if you can see my arrow, there's definitely one species there. And I can tell you that that's different from that yellow one, and that's different from that yellow one. And that one's actually different from that yellow one. And then all of the little dots in the background are sponges. And all of these white fluffy bits at the bottom are a different species, a different family of coral called lace corals. But my point is, in this one shot here, there's an enormous array of diversity. And one of the aims that we were trying to do was to get as many different species as we could collected by the remote operated vehicle so that we could try and work out some of the diversity of this area. Because visually from an image, sometimes things look really similar. And it turns out when you look at them close up, they're different. And this is exactly what happened in the case of this new species that I'm going to show you today. So we're in the deep sea and we've asked our extremely experienced remote operated vehicle drivers to pick us up this coral, because I didn't think I'd seen this coral before and we didn't think we'd got collected one. So there is the arm in the top left-hand corner, plucking it off the seamount, and it gets picked up and it gets put into a box on the front of the remote operated vehicle, and that box shuts. And at the end of the dive, which could be 18 hours later, we bring it up to the surface, and this is the front of the remote operated vehicle on the left, and you can see the boxes, and that on the right is what you call coral joy. Look how happy I am at getting that coral out of that box, right? So, and this is what we pick them out of the box, we put them into a cold water, because of course the water in the deep sea is very cold, and we want them to be very happy for as long as possible. So we put them into a cold water bucket, and we run into a cold room, and then we subsample them for genetics. We often subsample them for isotopes. We put some in formalin to make sure we can do reproductive studies on them. We take pictures of them and we ensure that they're beautifully preserved for hundreds of years to come. So when I go into museums, I'm sometimes looking at corals that have been there for over 100 years, and I can still, you know, investigate them and look at them and try and work out what they are. Now, so that was in 2011, remember? So I thought I'd ID'd them all. I thought I'd done a really good job. I described quite a few species in 2017. And then in the last few years, we've got money together to do some genomics, which is where you take markers from around the genome and you make a tree of life for these beautiful corals. And all of the things that I thought I described should have clustered together. Now, the circled species here fell out at a different section of the tree than we expected it to. So we went back to the original specimens in the jars and we had a look at them, and lo and behold, they were not what I thought they were in the first place. They were just a little bit different. And it turns out that this is an entirely new species. And this is what it looks like. So this is a new species of Thurella. It's a planar coral. It's one of those flat yellow fans that we saw. It has polyps, which are about 2 to 3 millimeters long, to give yourself an idea of how big they are. And the polyps are biserial. And there's a close-up of a polyp there. And if you were to dissolve all of those scales you can see on it off, they're individual sclerites. And it's the size and shape and orientation of these sclerites that tells me that this is a different species that hasn't been found before. So there are about 30 odd species in the genus of Thurella, and I've looked at all of them, and this isn't one of them, right? But I wouldn't have even looked at it properly if it wasn't for the genetics. So it's a nice mix of genetics, which is kind of very modern science, and old school morphological science of actually looking at these things, right? Now, this species, quite excitingly, we're going to launch on World Ocean Day to let a member of the public decide its name. So if you wanted to name a deep sea species, this is your chance. So yeah, you have to give it a good idea, think about what you would name it after, write it down, and we'll be making decisions about what this species is called in September. Now, the process of describing this species, we know it's new, but what we need to do now is actually take some beautiful pictures. So obviously there's pictures taken here in light that you can see, but the next step is to take scanning electron microscope pictures, which is where rather than using photons of light to take an image, you use electrons in a vacuum, like all sealed, and you get really granular, very detailed pictures. So this is an image of a species from the Southwest Indian Ocean that I described a few years ago, but you get the idea of what the type of images we need. So that's going to happen this summer, and then that species can be described, which is, you know, kind of exciting. So obviously, huge thanks to everybody for, you know, letting me talk to you about this today, but also obviously to the crew and the expedition who give all that time, and to the funders and partners who helped us get the JC66, which was the name of the expedition, underway. And that's yeah, that's how we're going to describe this new species.
Thank you so much, Michelle. That was extremely fascinating. Like, I am invested and I want to contribute to the naming and everything else. And I just found like it was so cool learning about, because, you know, we know so much about shallow water corals and not so much about deep sea corals. So
Yeah.
I really appreciate you taking the time to share more on what it looks like, exploring, you know, coming up with names, etc. And the map that you also showed, you know, showing how much we know, but still how much we still don't know, you know, all the gaps that exist in science, and then we're seeing all these different emerging threats. So that was a very nice visual, appreciate it.
Oh,
And
thank you.
yeah, I know we have our next speaker, Sadie, who isn't here with us, but Camille, I believe we have the video to play, so yeah.
Kia ora koutou, and hello to everyone. My name is Sadie Mills, and I'm the collection manager of the invertebrate collection at the National Institute of Water and Atmospheric Research in Wellington, New Zealand. I'm going to share with you today the early results of the Ocean Census Bounty Trough expedition, which set sail in February this year to explore an amazing deep sea region off the coast of the South Island of Aotearoa, New Zealand. Ocean Census is founded by the Nippon Foundation and Necton, and they're on a global mission to accelerate the discovery and protection of ocean life. And if you haven't already heard about them, I encourage you to jump over to their website later and watch their introductory video to learn more about their mission. Ocean Census approached NIWA to discuss the opportunity to explore in our part of the world where we would most be likely to discover new species. So we got together with colleagues from the Museum of New Zealand to Papa Tongarewa to compare our existing marine invertebrate and fish data sets to see where we had gaps in data and our knowledge of deep sea life there. This map on the left is a plot of all of the marine invertebrate samples held in the NIWA invertebrate collection, and you can clearly see the areas, particularly in the deep sea regions, where we virtually know nothing. For this expedition, we decided to focus on the large highlighted area called the Bounty Trough, just off the South Island of New Zealand. If you look at the map on the right, you can see the trough is about 800 kilometers long, from the canyons starting at about 600 meters deep off the coast of the Otago region, sweeping down the slopes to the many tributaries of the Bounty Channel. These start at about 1,000 meters deep and finally end up at the terminal fan of the channel in the abyssal plain at 5,000 meters deep. There are also seamounts and really interesting pockmark features that might indicate some kind of seeping kind of activity. And they're on the slope of the Bounty Trough. And these different habitats provide us this opportunity to survey these different types of environments that animals might live in and maximize our potential discovery of new species. There has been previous geological research, sampling and research in this region, but practically nothing is known about the seafloor biology of the region. This map shows the final route that our voyage took and all of the sites that we visited along the way. The table on the right-hand side shows the depths of some of the different sampling locations. And you can see those canyons and the sites at the start were around the 600 meters, and we worked our way deeper till our deepest location, which was just above 5,000 meters deep. The voyage was co-led by myself and Alex Rogers from Ocean Census. The team on board the expedition included 16 research vessel crew, 2 videographers from NIWA and Ocean Census, and 19 scientists and technicians from NIWA, Te Papa, Ocean Census, and several other Ocean Census partners from both New Zealand and overseas institutions. These experts represented the groups of fishes, mollusks, sponges, corals, worms, crustaceans, and echinoderms, and we also had seafloor camera and multibeam operators on board with us. Our sampling equipment on the research vessel Tangaroa aimed to capture animals living on and in the seafloor. We collected seafloor video footage with our deep-towed imaging system camera, and this can be seen in the photo just in the bottom left of the screen. An array of sizes of animals from tiny myofaunal animals living between the sand grains was collected from multicores and grabs, and you can see that at the top left of the screen. And larger fauna living on top of the mud or rocky seafloor was collected with a variety of sleds, trawls, and fish traps. You can just see a little video of the research vessel Tangaroa on the right-hand side of the screen as well. This clip gives you an example of what our towed seafloor camera sees when we first descend down to the seabed. Here is a school of Oreo fish over a sandy seafloor with Tam O'Shanter sea urchins. These are inflated deep sea urchins with a flexible, almost leathery test rather than a hard shell. 7 new species of Tam O'Shanter were described from New Zealand waters in 2013, and our taxonomist Owen is currently identifying the samples from this expedition to see if we've found any new species. Here next we have a clip of a seamount site where on the rocky seafloor you can see these beautiful large golden primnoa coral trees which form a habitat for a whole host of other invertebrates and fishes. The seafloor is covered in thousands of demosponges and the odd sea star, and if you look closely you'll be able to see other invertebrates. The seafloor camera, and here's some still images, gives us many first records of species to be discovered in the Bounty Trough, and the deepest records of species from around New Zealand. The still camera and the video footage is already slated for analysis for ecological studies and will be useful for a whole host of future research as well. A really cool example of the seafloor camera capturing deep sea animal behaviour and life can be seen here. This is a Taningia squid flashing its light organ at our camera. For reference, the laser points in the middle of the image are 20 centimetres apart. By the end of the voyage, we'd collected 17 hours of video, 6,000 seafloor images, and nearly 1,800 invertebrate, fish, and unsorted macrofauna and myofauna samples. These came from 107 stations at our 19 sampling sites. Immediately after the voyage, NIWA and Te Papa hosted a workshop attended by 30 taxonomists and parataxonomists from the Ocean Census network, internationally and within New Zealand, people from NIWA, Te Papa, and from elsewhere in New Zealand. Our sorters picked out hundreds of tiny organisms to put in front of the experts, who then identified, imaged, and drew the new species that they found. Experts have so far discovered more than 100 new taxa. These include new species of eelpout, more than 50 new mollusca, a new genus of black coral, a potentially new family of soft coral, and that's the 6-armed creature you can see on the right hand side of the image, 18 new and 8 potentially new amphipods, 19 new bryozoans, 1 new stylasterid hydrocoral, 2 mysids, 2 new shrimp, and 19 nematodes. And that's just so far. There are also new depth of range extensions for a variety of taxa, and additional specimens of rare and known but undescribed taxa. And there's still a lot more work in progress. A really big thank you to our videographers, vessel crew, and science partners for the images and video used in this presentation, and for making the voyage the huge success that it was. I'd like to acknowledge the Nippon Foundation Nekton Ocean Census Program for supporting the Ocean Census Bounty Trough expedition. We look forward to caring for the collections to document and describe the biodiversity of this underexplored region of Aotearoa, New Zealand. NIWA and Te Papa are custodians of the samples and will make them available for a whole host of future research. Thanks very much for listening today.
All right, well, that was from Sadie. Very interesting. I kind of wish she was here. I have questions. So much that we're learning and discovering, and just getting a deeper insight into what goes, what takes place when you go on these expeditions. Like I keep saying, it's just really cool, really fascinating, and I'm just 100% invested. And I feel like maybe within the next 5 years, I have to find a way to join an expedition of some sort. I don't know how, but we'll make it happen, right? And moving on to our last speaker, we have Salome.
Hello. Hi. Oh, do I just go for it? Do I dive right in?
Yeah, go for it. Go for it.
Oh, awesome. Okay, here we go. Just want to say how it's great to be here with friends, meeting new scientists, and also to have you here, Khadija. I'm also from Trinidad, so I just love hearing your accent.
Yay!
Yes, Caribbean represent. All right, I'll take us away, if I can make this work. God, I've done this so many times, and here I am. Okay. Share screen, and can I just get someone to say that they can see my screen? Awesome. Thanks for the thumbs up. Yeah, so I'll be talking about some of the deep sea discoveries we've made in the Galapagos. But before I start, I want to acknowledge that I'm presenting from the unceded territories of the Musqueam, Squamish, and Tsleil-Waututh Nation here in Canada, and it's a privilege for me to work and study and do my research here on their lands. And as they said earlier, I'm a PhD student, so I feel very honored to be amongst these senior scientists presenting here. And I'm also an affiliated scientist of the Trans-Saharan Research Station, where I lived and worked for 4 years, and that is in the Galapagos. So I'll get right into it. I have had the privilege to develop a deep sea career on this tiny archipelago in the middle of the Eastern Tropical Pacific Ocean. And the Galapagos is a province part of Ecuador. It has people living on it. And here's a close up. And the dark line you see is the Galapagos Marine Reserve, which is one of the largest and oldest marine reserves in the world. And Galapagos is famous for Darwin and its giant tortoises and the land species, but as all marine biologists agree, the most incredible part is in the ocean. And it's just one of these fascinating places where we see tropical and cold water species living together in the shallow reefs. And it's just like, for anyone who's a marine biologist, that's mind-boggling. What is a parrotfish doing next to a penguin? And you will see that in the Galapagos. You'll see kelp. Well, you won't see it often, but there is kelp there, and living side by side by tropical reefs. And one of the reasons why this happens is it's just in such an exceptional oceanographic and geological setting that it's this archipelago that just rises up in the middle of the ocean where there's nothing else, in the middle of 3,000 to 5,000 meter depths. And it rises up, and it's at the confluence of 4 currents that are different temperatures. And this is why you can be in the Galapagos and jump in the water, and it's 27 degrees in one place, and then it's 17 degrees in another. And if you look down, it looks like you're in the Caribbean, and then suddenly it looks like you're in California. But the other reason why this is also interestingly mixed up is because we have here a really complex topography in the deep. We have many seamounts, small and large. We don't know exactly how many because we're still in the works of mapping everything carefully. This is just a kind of cartoon image based on the best topography that there is. And what you can see here is that we have these really large, what technically are seamounts from 1,000 meters going up. But we also have these smaller, what I call small seamounts, but technically they're knolls or little hills on top of the Galapagos platform. And what is so awesome is that not only do we have various seamounts together, small and large, but they've been protected. There's been no industrial fishing here. There's been no activity. So we have an opportunity to study deep sea ecosystems in a place that's close to pristine. And we always suspected that there's probably incredible deep sea life given as the discoveries of deep sea corals around the world, but we weren't for sure. And only 10 years ago, we launched the Deep Sea Research Program at the Charles Darwin Foundation. We're a tiny team, but we have gotten amazing support collaborating with different institutions and universities who come with incredible technology to take a deep dive and see what's living down here. And this program is currently led by Stewart, and I was part of this team for 4 years. And the expeditions that I've been working closely with and a part of, mostly on the Ocean Exploration Trust, on the Nautilus, but also where I got to go on the Atlantis and the Falcor. And these vessels did an incredible job at helping us map more where we have these deep sea spaces. And that's one of the key things of exploring the deep, is actually mapping it first. We actually need to map so much more of the ocean. It takes a lot of work. And where you see the more colorful, the more brilliant colors, those are the places that have been explored by these expeditions and were also mapped. And most of my time, I actually learned about the deep sea from the Nautilus expedition. They explored 4 seamounts in the Galapagos, and they confirmed that we have incredible habitats, but mostly we have these cold water coral reefs that Michelle and the rest have been highlighting about, that these are habitat formers that host a myriad of species. And here we can see this is a hard coral, Sclerenctinian, and on it we have some squid lobsters. Around it we have the gorgonians as well. And what this just affirmed was that we have these cold water coral reefs in the Galapagos, and that we now have an opportunity to study what these are like for the Eastern Tropical Pacific and find out if how they're connected with the other cold water reefs in the Eastern Tropical Pacific corridor. The other things that we know, we also found other important distinct communities, including what we call coral gardens, which are made up of gorgonians, sponge gardens. We had a mix. We also noticed these habitats formed by echinoderms. So we had these ophiurid beds, just like hundreds and thousands of ophiurids, beds of crinoids. But yeah, the one that really struck with us were the cold water reefs. And one of the reasons is the depths at which they are. They're between 200 and 600 meters. And in the Eastern Tropical Pacific, sorry, my slides have been mixed. In the Eastern Tropical Pacific, we are in a place, this is a map of the oceans globally, and the blue parts show you the parts of the world where between 200 and around 600 meters that we have an oxygen minimum zone. This is a zone where oxygen is less than 2 milliliters per liter. And what this essentially is a hypoxic environment. And these exist in parts of the world, like in the Indian Ocean, and the Eastern Tropical Pacific is known for it. So we were really curious to see how much life is in this depth band in a place like the Galapagos, which is why when we found that there's this in exactly that zone, we were just like, okay, this changes our paradigm of how we understand the distribution of complex habitats in the deep sea. So the deep sea keeps on reminding us of how little we know and how much more we need to dedicate science and efforts to understand it. This is part of a data set that I analyzed, and here we have a vertical profile of the substrate type and then the habitat type on it. And here again, it shows where the cold water reefs, this pink band that I put across is here exactly where the hypoxic environment is. And we are really interested in understanding what adaptations have these corals evolved into actually withstand these hypoxic environments. Or maybe their tolerance is different than what we expected. This again shows this, and from the last expedition, we did so many CTD casts. Oh my God. But it was well worth it, because for the first time, we've got beautiful profiles of understanding where this oxygen minimum lies across the archipelago. So these are important questions that we can answer now that are uniquely situated in the Galapagos. recently I got the opportunity, I felt like I was living in a virtual reality because I've been just looking at hard drives of data. And then I got the chance to actually go down there and see these amazing ecosystems myself. And hope you recognize Michelle here. I had the amazing experience of not only being in the sub there, but being on this ship, stuck on the ship with Michelle for 6 weeks. It was amazing. I learned so much about corals, as you can imagine. And it was actually during one of Michelle's dives that we discovered this massively expansive cold water reef. And here is what you can see. This species is called Madrepora, possibly Madrepora oculata. And this is actually a cosmopolitan species. So it's really interesting that it's living here in the Galapagos, far away from its peers, but also in this oxygen minimum zone. And just notice how it just goes on forever and ever and ever. And we learned that it's covering more than 1 kilometer squared of habitat. And this is something that we were able to go back to when we came back on the Falcor. And I also had the chance to be on one of these types of expeditions where we're not using a submarine, but this massive ROV that you can see here. And we were able to fully map where this habitat was, and we're calling this the croissant, or el cacho de coral in Spanish. And the reef was all along this top here, and it was very, very expansive. But the other interesting thing was that on the same expedition, we went to other sites and we were finding more and more of these reefs. And yeah, from the Nautilus, where we only did 4 dives, we found a few, to finding so many more after doing, I think we did 26 dives with the ROV. And we are now pretty certain that the Galapagos deep sea is just covered in cold water reefs. And there's just so many more questions, really. These discoveries really just like highlight that we need to do so much more than we originally thought. And not to mention from collecting all of these samples that we do when we're out there, the work goes usually on for another 2 decades until taxonomists go on to help identify and find out if these are new species, describe these new species. And this is something that at the Charles Darwin Foundation, we've been, a very small team, have been working really hard on. Once we get these images, we start to work on doing an inventory of everything that we saw. Half of the time we're categorizing it because we don't know what things are. For example, there was this sponge that looked like a coral. We're calling it the sponge with the coral complex. We've been working with over 25 taxonomists all over the world who are kindly helping us identify these. And we've published some of the findings, but we're also working towards providing the first what's the name for it? Deep sea guides for the Eastern Tropical Pacific, which is going to be critical as we continue exploring the mountain range, the seamount mountain range that exists between the Galapagos and the Costa Rica, where we have a chain of seamounts. And lastly, so while I was living in my virtual reality of just studying deep sea habitats on a hard drive, I was like, I can't wait for one of these vessels to come and take us to the deep. Surely there's other ways to do this. So I realized that there's actually cheaper ROVs down there. They can't go as deep, but they go as deep as 200 meters. And we're just like, there's shallow seamounts right here on the platform, as shallow as 100 meters. So why don't we start exploring those? And in fact, they are really important to the Galapagos Marine Reserve because these shallow seamounts are actually found by the fishermen, which is where they tend to fish. This fish you see down here, the brujo, is the most common fish you will see being sold freshly to eat if you go to the Galapagos as a tourist. So I was like, these are probably really important knolls, and they probably provide a lot of habitat where these fish are being fished. So why don't we figure out how to go down there? So I collaborated with Victorero, who provided us with these smaller ROVs. They can only go down comfortably to about 300 meters. And it even has a little claw, so we could do a little bit of sampling if we wanted to. And the setup is a lot more affordable. It's affordable within a budget of a PhD student. And so this is a little boat, and this yellow cable here you see is a tether, and this is becomes super annoying when it gets entangled, which is why we've got it in a figure of 8. And I thought I was gonna find cold water reefs or Gorgonian gardens, and instead what we're looking at here is a kelp forest. So we accidentally discovered a brand new kelp forest to the Galapagos, and now I have the second love of my life, which is seaweed after deep sea. And that's actually what I decided to do my PhD on, is characterize and understand this mesophotic habitat that is not in the shallow. So we didn't know it existed just because it's a little deeper than our scuba diving capacity. And I had the chance to also go in this sub here to go and see it, because in the ROV you kind of get just like goggle vision, or and again, football fields of this kelp covering the top of this shallow seamount. And for the last 2 years, my existence is this. I'm just looking at video transects that I did. I'm using stereo video methods. I am measuring stipes to understand the population structure. I managed to collect some samples, and I'm aging these kelps. And if you're interested in learning more about the mesophotic kelps of the Galapagos, we published a paper last year, so it's out there. And that is my time. But I just want to remind you, like, if little me is just dropping a small ROV and going a little deeper than usual, and I'm discovering a whole new ecosystem, I think it's a stark reminder of how little we know and how much more is in our deeper ocean that needs to be discovered. And I thank you.
Thank you so much, Salome. And once again, Trini representation. So happy about that. Yeah, and just the fact that you shared that ROV with us, that is something that we at SOA have been discussing for a while now, getting one of those to be able to go and do some exploration, see what we find, just like how you found that kelp farm that you did not expect to find, So yeah, very interesting. The Galapagos is one of the biodiversity hotspots, so I could just imagine what it's like studying there and just always having something new to find. And I feel like we have a very common trend throughout all the speakers, which is that we're discovering so much, but there's still so much that we absolutely don't know, and how much time that it's going to take to keep uncovering all these new species. So thank you all for these lovely presentations. We're going to jump straight into the Q&A. And I think one of our first questions, and feel free to jump in as you see fit, would be, how do we get people to care more about the deep sea? And then what are some of the threats that the deep sea faces? So no rush, no pressure.
That's a big question. Yeah, there was a wonderful paper from Alan Jameson's group a few years ago, just talking and discussing about how, because the deep sea is a long way away from everybody's everyday lives, and I find this astonishing. And it turns out us deep sea geeks are actually in the minority. Most people don't like deep water. So it could be considered a pretty hard sell, but I think doing the type of things that sharing the type of video that Salome was just sharing, the showing people the deep sea, I think that's step one, right? So understanding is kind of step one to acceptance and caring about this. And as Lissette said at the beginning, like, it is such an important ecosystem, and it does actually play quite a big role in all of our existences because of its role in global processes. So again, getting that understanding across, it's I think education is probably step one in terms of making people care.
Yeah, fully agree with you. Anybody else want to jump in, or you want to go to the threats that the deep sea faces?
So many.
I'm happy also to jump into the deep sea threats. Basically, at the moment, the biggest threat to the deep sea is deep sea fishing. It's been going on since 1950, so we're coming into 75 years of deep sea fishing. And basically, the biggest, most destructive practice is bottom trawling in the deep sea. We're obviously talking a lot about mining at the moment, climate change, and all of these other things that are happening or will happen. But what is happening right now is this habitat destruction. And it takes a really long time to recover if the system will ever recover. But we've seen that it takes at least about 40 to 50 years for these type of corals that Michelle was showing, that Salome was showing. It takes a long time to recover. And basically, I think it's one of those things that needs to change. And I think it is about education, as Michelle just talked about. I don't think, unfortunately, we are as educated, and policymakers are not as educated on the topic as they should be, because obviously otherwise it would be banned, because it makes no sense to, use taxpayers' money to go and bulldoze the oceans. All of these deep sea fisheries are subsidized as well. So it's an important point to take into account that it's not a profitable business in its own. And I think this is the biggest current threat to deep sea systems.
Yeah, I just wanted to add that one of the things that I find threatens it is the challenge of managing deep sea ecosystems, and that is what happens when, given that most of the deep sea is in high seas and parts of the ocean that don't particularly belong to any country, it means that this belongs to no one and everyone, and managing these spaces and getting countries to agree but also just care is very challenging. And yeah, I think that's also got like policy and agreeing on the policy and not allowing those who have interests that are motivated by money, which is the deep sea mining companies, to get priority over whether they can explore and exploit these sites. I think is also one of the challenges, is that we are working with ecosystems that are often in no man's land.
Yeah, before we wrap up with closing remarks and calls to action, I just have one fun question to wrap up, this deep day webinar, and that is for each of you to tell me your favorite deep sea species and why. In like 35 seconds, 45 seconds.
I'm happy to go first. I think my current favorite species is yet to be named. It lives in hydrothermal vents under the Arctic ice at 4-kilometer depth. And basically, we managed to access it. We were the first team to ever dive under the ice with an ROV in the Arctic. It's an amphipod. It looks like a little shrimp, basically. But it's very unique. It's one of the first fauna ever to be found living on hydrothermal vents under the ice.
That's really cool.
You have to top that now.
I know. Like I can't top that. No. My favorites are I really like Dumbo octopus for no other reason than they just look amazing. Like with the little flappy things on the top. Like how are they getting through life? I just find them I just really like the way that they look. That would be mine.
Mine would be the basket star, which are these ophiuroids that have these curly legs that remind me of my hair, and but then at the end of each of their curly legs they have like
Little
what
arms.
looks like little hands, and they're just I don't know, they're just adorable, and they just look really needy. They're just like hanging on with everything they can to corals,
And they get so big as well. They
yes.
can be huge.
Yeah, that would be my one.
Victoria.
You got a favorite?
Oof. I would go for the newly discovered little sea toad. I don't know if you've seen a picture of it. Looks like it has sort of crocheted skin, and it just looks fluffy, and it looks like it shouldn't be very, very deep underwater, but it's very cool and bright orange. So it's just very cute.
That's amazing. I'm gonna have to Google these.
Nice. Michelle, I believe you said Dumbo octopus, right? Yeah, that's my favorite too. I absolutely love them. They're just cute. That's one of my Zoom backgrounds, but we could go on and on about
Nice.
these
All right.
creatures. But I know that Camila has some closing remarks, and then I would wrap up everything or some closing key messages. So go
Yeah,
for
hi
it.
everyone. As we were talking before about some of the things that we can do to protect the deep sea, the Deep Sea Conservation Coalition obviously is asking for a deep sea mining moratorium and to stop deep sea bottom trawling. If you want to learn more about these threats and what we're doing specifically to protect the deep sea from these threats, you can go to our website. So I'm just gonna post it in the chat. Yeah. Thank you, Khadija.
Yeah, so I hope you guys enjoyed this webinar. I know I did. I was, like I said, 100% invested in all the presentations, and I can't wait to learn more to follow up, and stay tuned for the coral naming competition that will launch on World Oceans Day. Very excited about that. I mean, I'm gonna submit. I feel like I'm gonna win. I'm just putting it out there, but like anybody else, feel free to contest me. That's fine. And like Camilla said, we have all these threats to to the deep ocean, and we really need, as much people as possible being informed, sharing as much information as they can with their peers, and really pushing and getting governments to join the call for a moratorium on deep seabed mining. And all the other 500 threats that the ocean faces. And yeah, it's just very much overwhelming. But as we saw today, the deep ocean is absolutely fascinating and incredible. And there's still so much that we don't know about it, which is definitely why we need to invest time and energy into protecting it and discovering all the wonders and all the ecosystem services that it has to offer us. And on that, I feel like we just 1 minute to closing. So do I ramble for 1 minute, or do I just wrap up? But I hope you guys enjoyed. This webinar was done by DSCC in partnership with Sustainable Ocean Alliance. You could also look out for Sustainable Ocean Alliance on LinkedIn, Instagram, everywhere else. And yeah, happy deep day, everyone. Keep posting those deep sea messages and keep remembering to be connected and love the deep ocean. So on that,